So it's my pleasure to have Mo Lotfollahi here. And Mo recently, very recently, has started a group, as a group leader in the Sanger Institute. But I've just chatted to him, and he's also keeping his Helmholtz Munich affiliation as well. But he is going to be based, I guess, in Cambridge area, in Hingston. And it's really exciting to have him over this side of the channel, I guess, working there. And we're going to hear about machine learning to understand single-cell behavior. So over to you, Mo.
Awesome. Yeah, thank you very much, Magnus, for inviting me, and it's great to be here. So I'll talk about multiple problems in single-cell biology from different aspects that could be addressed using machine learning. And I think it was great because in the previous speaker, it was already a nice introduction to single-cell biology. So I don't have to repeat many things. But I'll start with basically showing this fruit analogy that we have for single-cell biology, that where we have started. So there was like bulk transcriptomic, where people actually show it with mixed, it's like basically smoothie, where you don't know which fruits were there. And then and then it was like single-cell genomics, where basically the fruity now got decomposed into individual fruits and smoothie. And then we have spatial biology, spatial genomics, where now we actually know organization of cells. And then you can see this beautiful chart there as an analogy there. So, and then another thing is, there is this consortia called Human Cell Atlas. I'm just trying to lay out why and how single-cell biology kind of can help to understand health and disease. And then what people are actually doing is, as part of like Human Cell Atlas, many collaborators in universities and research institutes, they try to basically build a table of cell types, tissues, similar to the one that we have for elements, basically chemical elements. And the ultimate goal here is to use, so single-cell biology, single-cell genomics is basically is merely a tool to understand behavior of cells in a very, very high resolution. And my research is closely tied to what Human Cell Atlas is imagining, basically. Building a comprehensive reference in health and disease. And so the first question is, can we actually use all of the existing data out there to understand the difference between health and disease? And then understand how we can transition between different states. So basically health and disease. Can we find a treatment, or can we push one state to another? So basically modeling perturbations. Perturbation could be drug treatments, could be CRISPR knockouts, could be even disease. And then taking our understanding from the tissue and also how perturbations are affecting cells, then mapping that to population genetics and population biology. So where we try to now understand how this variation in cells, tissues are mapped in population. And since we're dealing with like tons of data, lots of like kind of complicated data, then I think like ideally we want to have like a digital twin for every tissue that is shown here, meaning that we need to have like an in silico model where we would be ideally to use that to understand, perturb, and then map it to population. So using in silico model, machine learning, computational biology to further understand this. And so in the previous slide, I mentioned this Human Cell Atlas project. And when the word atlas comes up, So it basically implies something big. And it means that we're collecting data sets across multiple sources. And in the previous talk, it was mentioned that there is something called batch effect. So here, I just want to repeat that once more and then show you that as you see here in the first row, so you'll see 4 different data sets colored by 4 different colors. So each dot is a cell. And this is a UMAP representation, so basically like a 2D representation of the data. At the second column, then you see same data set, but colored by different cell types. And as you could see, the cell types are scattered based on the source. So if you look at these alpha cells in the second row, then you'll see they're scattered all over the place, whereas they're biologically similar, so they're the same cell identity. So ideally, we need to have a representation like this one, where all alpha cells are now clustered together in one cluster. And this is the representation that you could use for downstream analysis. So during the talk, so basically go back to like the first slide. I'll show you methods that help you to understand this heterogeneity across patients and population. And then we zoom in into cell states and cell clusters. And we further zoom in into individual cells and see whether we can actually model their behavior. And then one last piece of this is we can actually look at the morphology and the spatial organization of cells. So I'll show you machine learning, ML, statistical methods along this spectrum of resolution that can actually help us to understand health and disease in different resolution. And the first thing is I'll start with a notion which has been taken up recently in analysis of single cell data, which is you don't want to repeat all the steps that was mentioned in the previous talk. So clustering the data, talking to an expert to see whether they give you some marker genes or validate those marker genes to basically identify these cell states. It's a very lengthy and exhaustive process. It takes months. I've done it like 4 times and you don't want to do it. So the question is, can we automate this process? If we have a large scale data set for heart, so integrated across all data sets out there, we've built like this heart cell atlas. Can we use this heart cell atlas to map new data and then transfer information from this reference to the query data set? And these information could be cell type annotations, could be continuous information like missing modalities and etc. And so using a reference to analyze a query is not a new concept. So people have been using it in reference assembly, reference mapping, where we basically have a reference genome, and whenever we get a new sequence, then we try to map it to this reference genome and see the similarities or differences between the query and the reference. So ideally, we would like to have similar thing here, where we have a reference building algorithm, so where this reference building algorithm build a reference, and now we have a reference mapping algorithm that takes a new data set and map it to a reference and allows it to transfer information from the reference to the query. Why is it good? Because it's very expensive, A, computational, B, a lot of effort to build a reference because you need to harmonize the data, you need to, it's a lot of work. Yeah, it's just like building one of those atlases is like a 3 years project with like 50 people. And so you don't want to do it again. So the idea is there are a bunch of challenges here that it basically doesn't allow you to map data easily from a query to a reference. So the first thing is there is batch effect. So between the query and the reference, there is batch effect. So you cannot just concatenate these 2 data sets and look at them together. So you need to basically resolve and correct the batch effect. B is if you have a reference, then you need to have a way to send your data to a collaborator. If you think about a reference and now everybody wants to map that reference, then you need to kind of come up with algorithms where it's actually distributed. So you don't have to be worried about sharing your data, so and also preserving privacy here. And another important thing, I told you about the computational efficiency and like how challenging it could be to build an atlas. Then with the reference mapping, things could be quite faster because you don't need to rebuild everything. So it's just mapping one data set to another. And finally, it allows to make all of the existing data sets more accessible to everybody because you don't have to be a big lab to make this reference atlases and then have a lot of expertise. So any individual or smaller labs could have access to these references and they could leverage this. And this was basically our motivation. I think that was part of my PhD where we proposed a method called SCArches to basically allow reference mapping for single-cell biology. And this method is based on leveraging state-of-the-art generative model in single-cell genomics to correct batch effect. So I'll separate it into two parts, reference building and reference mapping. We assume that we have a reference building algorithm. And in this case, the reference building algorithm is a conditional generative model, so conditional VAE, where you input the model. So the input of the model is a gene expression for that cell and also a condition, so a conditional variable, which is the source where this cell came from. So the batch, the experiment, etc. And so this is the first phase. And then we assume that we are given a reference, a pre-trained model based on a reference. And then what we imagined at that point was basically we tried to push all these pre-trained model. I don't know how many of you have heard of like Hugging Face? Okay, so that's basically like a repository for large-scale machine learning methods where models where people can download pre-trained models and use it for downstream. And the idea is to do something like this. So push pre-trained models. And then whenever you have a new data, then you can perform transfer learning by mapping disease data on the top of the healthy atlas, or you can do transfer learning between species or organs or studies. And the idea was quite simple. Once you have the query data set, you want to keep the weights of the model fixed. So you don't want to change the reference because the reference is like a map. So you need to fix the coordinate. At the same time, you want to correct the batch effect. So the idea was we kept all the weights frozen in this conditional architecture, and we only fine-tuned specific weights that are connected to the conditional embedding in the inputs of the model and also in the latent space. So these are the nodes that are responsible for informing the model that this cell is basically from study 1 or study 2 or batch 1 or batch 2. And the idea was to only fine-tune this. With fine-tuning these weights, then you keep the internal weights frozen. So you don't change the coordinate, you don't change the reference. So if you map the reference data again to this, to the latent space of the model, it will be same. At the same time, it's fast. So you're optimizing, I don't know, like if you have 2,000 genes as input, you're just 2,000 parameters. It's as fast as running a logistic regression or something. So you can run it on your laptop. And finally, it's order invariant. So it doesn't matter if you have a pre-trained model and if you add a new data set and a second data set, the order really doesn't matter. So you could iteratively update this. And this is also very, very close to the notion of like lifelong learning and continual learning. Yeah. Mm-hmm. Oh yeah. So there's a trade-off here. There is a trade-off that you could actually, identify the right weights. So the question is whether you actually need to identify or fine-tune these specific weights versus first layer, second layer, or you search through the whole architecture and find the relevant neurons to update. So in this case, all the neurons that you update here, it's not going to change the input. So the input is basically fixed, so the input representation. So that's the first guarantee. And the second guarantee here is, like another trade-off here is the computational efficiency. So we also took that into account, that you only fine-tune those. And so the guarantee here is like, it's a linear operation here. So you don't change the weights of like previous network, and you only connect it, assuming that these are the weights that inform the model about the condition. So if you subset to these, then you guarantee that you don't change it. And so disentanglement happens naturally because, I mean, we wrote it in the paper. This is similar to writing a linear model in that case. So you have like the weights of this conditional model times input plus a bias. So it's completely disentangled in that sense. You're not changing other neurons. So we have, I'll show you a few examples. Yeah, and then you see, you get a scale. So, but we're thinking about like 100 data sets right now. Yeah, and like millions of cells. So I'll show you one example how this pipeline works. So the first one is building the reference. So there are like multiple methods out there. You can use them like SCBI, ScanBI, and we also developed a few that you can build this reference. So this is unintegrated reference. So you see batch effect. So the first row is batch, colored by batch. The second row is colored by, it's colored by cell types. And this is the first part. So we used a method called TRVE. So that was also part of the method that I developed for the reference building, but then, and it's generalizable. So we also showed that it works with other methods. So this is the first part. So the pre-trained model on the reference. Now we added a query data set. So this, the query data set is CellSeq2. And what you see is Pancreas Atlas, and CellSeq2 is completely different technology compared to the Droplet or SmartSeq technologies where you see where the reference was built based on. And so in this example, we basically simulated a scenario that you might even have novel cell types in the query that might not be present in the reference. So we left out all the alpha cells from the reference, and we left them in the query to simulate this scenario. And as you could see, the alpha cells are not merged with the existing cell types. So this is merely based on UMAP, but like we further quantified this. So this is unseen in the reference, and then you can iteratively update this without destroying the reference representation. And in this case, we added a second data set. So we start from 3 data sets to build a reference, and then we iteratively added more and more data sets here. Okay. Another example is, once you build a reference, the reference could be based on multiple modalities. So in this case, I'll show you an example where we build a reference, where the reference included both single-cell RNA-seq and also site-seq data sets. So site-seq is another technology. Was it mentioned in the previous talk? I can't recall, but like, so site-seq measured both gene expression, single-cell RNA-seq, and surface protein abundance. So you have 2 modalities here. And what we built here was a reference with both of these modalities. And the idea is, can we use this approach to map a new data set where you only have one of these modalities, so only gene expression? So map it to this multimodal reference, and then impute all the missing proteins that you don't have in the query data set. So this is the query data set. It's a 10x data set where you only have RNA-seq. And this is, and just to mention, this is based on a reference building model called Total BI, where it's basically an established method to integrate single-cell RNA-seq and so site-seq data sets to, for multimodal data integration. And we applied ST-arches on Total BI in this case. And what you see here is the integrated representation of the reference and the query. And what you see on the right is basically imputed protein abundance for the query data set, which was only single-cell RNA-seq. So the idea is this transfer learning could not just be on like cell type levels, which I'll show in the next slide, so discrete information. It could be also continuous information that you could further impute. And I'll show you another example, which is, I think, the most use case, mostly like the community really picked this up, where they try to build a healthy reference atlas, and they want to basically map a disease data on the top of this healthy reference. since the model learned to correct for the batch effect in the healthy data set, it would be able to correct the difference between the disease and healthy that only exists in the healthy data set. So if you have a disease data set, the effect of the disease will be remain. So the model by design cannot correct for that. So if you look at integrated object of the reference and the query, then you'll see cases where one cell type might not be integrated, and that might be the disease effect or the batch effect. So this is like a scenario that we explored in the paper. In this case, we built a healthy reference from 3 different tissues. So you have bone marrow, lung, and PBMC. So we integrated, if I remember correctly, it was like 300,000 cells. And then we mapped a query data set. So this is lavage data set. So it's like a proxy for having, for obtaining lung cells without really dissecting the lung. And then we mapped this data set on the top of this healthy reference. And in this data set, we had data from 14 different patients. And these patients were diagnosed with COVID. So a few of them had severe COVID, and then a few of them had moderate COVID. And then we also had some control healthy samples. So after mapping this data set, then we sought to basically explore which cell types map to the reference and which cell type did not map, and if this is batch effect or this is disease effect. And we saw a big difference in a group of cells called macrophages. And so I'm not going to go into details here about macrophage biology, but what we observed was there were a group of macrophages in the query data set that were not mapped in the reference, into the reference. And that's what you see on the right. So, and there were a group of cells from the query that mapped to the reference. And indeed, those were the macrophages from the healthy patients. So these are tissue resident alveolar macrophages. So these are the macrophages that actually exist in the tissue. And the ones that were not mapped to the reference, they were monocyte-derived macrophages. So these are the cells that they don't exist in the tissue, and they're transferred to the tissue once you have disease. So they're like a novel phenotype that happens to be only in disease. And then we validated this with the help of like lung biologists to see whether this is actually a novel state or not. And we observed the same thing with CD8 T cells. So there were like some CD8 T cells that were only specific to disease, and we could identify them using this approach. Okay, so that was one example to show off, again, why and how you could use a healthy reference or a reference data set. And now more and more of these references are coming. So people are building these large-scale data sets. And one effort here is actually now published in Nature Medicine, it was last week, to build a human lung cell atlas. As part of that, a group of people, so this was led by Lisa Sikkema, a PhD student in Munich, where I think she worked with like 80 other people for 3 years to build this resource. And they started from a core reference where they integrated many data set to build a reference with 500,000 cells, which they call core HLCA, Human Lung Cell Atlas. And in through 2 years, they iteratively updated this reference atlas using SDRHs. So in the end, they extended this reference atlas to 2.2 million cells across 445 different individuals and multiple diseases. So you could see the distribution of samples in this case. So there were like multiple diseases, carcinoma, IPF, and COVID-19. And you could also see a representation of the core reference where they started and how they iteratively updated this reference atlas. So one thing that they explored, yeah, sorry.
In the latent space. Is this the latent space?
This is the latent space, yeah.
Do you have that here? I mean,
Well, that's for the causal reason, yeah. So you need to basically, if you have the identifiability issue with the causal structure, here we don't assume any causal structure. So so it's like a latent space we only corrected for the batch effect. There might be some, well, it depends. It's like designing a linear model. So what you want to correct for. And if you have a good knowledge about what you want to correct for, then you might end up having a good latent space. So if you overcorrect or undercorrect from covariates, so if you correct for disease, then you might not see disease effect at all. If you correct for technology, then you might, then that's actually ideal. But there might be like hierarchical batch effects. So if you have like one sample from one patient and that patient has like multiple samples. So the question is whether you correct for the patient level or sample level, or you could just go down in the hierarchy. So there is essentially, there could be that you could identify this as like identifiable problem because you want to identify latent variables and latent co-founders, which you might not know because, I mean, that's how it is. But you have some knowledge about the existing ones like batch, disease, age, sex, and all the metadata that you might have, and you could correct for them. But you might overcorrect and undercorrect, so this is actually, that's why you need metrics, and then there are people who are actually designing metrics to check whether, because there's a trade-off here, the trade-off between integration and preserving biological variation. And they're not always, you can't optimize both at the same time. Yeah. Okay. So in the paper, they wanted to showcase how can you use a reference atlas to analyze downstream data set? Can you use a reference atlas to identify subpopulation automatically? And this is what they use in the Human Long Cell Atlas effort, where they build this huge atlas, a comprehensive atlas, and they map data to this reference atlas. And what you see is one example where they mapped a completely new data set into the reference, and then they wanted to automatically identify whether there are new subpopulation in the query and not in the reference. So what we did in the original paper was just like, we clustered the data and we identified, okay, these two didn't integrate, and then we had some expert, and then I had to do some digging. But like the idea was like, can you actually make this more scalable and automatic? And the idea was to train a KNN classifier, a very simple KNN classifier on the latent space, where you want to transfer cell type annotation from the reference to the query. And this KNN classifier is aware of the batch, is aware of the uncertainty. So you can basically calculate some super simple distance-based uncertainty, where for every cell that you predict the cell type annotation for, you have a value of uncertainty. So the idea was, and what you see here is basically the projection of those uncertainty values for the cell type annotation predicted for the query data set. So you see some regions that you have high uncertainty. And what they did was basically look at this uncertainty values to see whether it's actually calibrated, whether it's meaningful or not, meaning that if the cells with high uncertainty were actually wrongly annotated by the method. And indeed, that was the case. So for the cell types that were that had a high level of uncertainty, they were actually misclassified by the classifier, and they were indeed the cell types that were not present in this in the reference. So they were like new subpopulation that were only present in the query. It was like a super simple method for outer distribution detection. And I like another example. So these are not my papers, but like I like to show this one, which is also quite fascinating in terms of biology. That this was also published in Cell this year, where by a group of researchers in Australia, where they built a single nuke resolution transcriptome reference for human prefrontal cortex. So what they did was they integrated across multiple gestational age and brain development, and they wanted to study how the brain developed from basically very early toward childhood and adulthood. And what they did once they build this reference, they mapped different type of data to compare the query data to the reference. And in this case, the query datasets were basically datasets from different ages, so gestational ages, to see whether some cell types are actually preserved in development. They also map in vitro data, so organoid model for brain mapped to the in vivo reference to see whether the organoid model was accurate and could recapitulate different cell types and cell states, which happens to be in the in vivo data set. And they also formulated, so these are all like, so you could check the papers, it's quite interesting. What they also did was, because they had the exact age of each cell based on embryonic date, then they formulated a kind of like age prediction problem from the reference to the query. So for the cells that they didn't know the exact age, but they had a comprehensive reference, they could basically transfer single cell age to build like a single cell aging clock, what they call it, which is also interesting. Okay. The next question, again, along the same line of building references, using these references, is can you actually build a hierarchy of cells? Meaning that if you have 3 data sets and they were annotated by 3 different collaborators, so one in Australia, one in Munich, one in Cambridge, with different expertises and different cell type annotation, can you automatically harmonize those data sets without integrating them again and then clustering and going through the whole pipeline? And B, can you, once you would be able to build a hierarchy, so basically if the annotations from the first collaborator were subsets of annotation of the second collaborator, can you A, build this hierarchy, and B, update this hierarchy with new data sets? So that will basically allow you to identify subpopulation and even put them in a hierarchy of groups of cell types. So this is a big thing in bioinformatics and computational biology. They're like a group of people that are like lifetime work on building cell type ontologies, and this is actually a big interest. I was not aware of that before, but that seems to be a lot of people actually trust in these ontologies, which is sometimes debatable. But so the idea, then we group up with a group of people, and so Ahmed Mahfouz, he's a PI professor and PI in Delft University in Leiden, and a PhD student there, Lika, who spearheaded the project. And the idea was to basically build a hierarchical model based on SCRs. So once you have the reference, then you try to build a hierarchical tree, and then upon the addition of the new data set, you try to update that tree. So I'm not going to go into the detail about the method, so it's called tree arches, but I just want to show you one example that how tree arches could help you to automatically, this is again along the same line of building references, identifying and reusing, identifying new subpopulation and reusing them. In this case, we built a cross-species atlas, and what you see is basically the starting tree. The method is super simple. So it basically tries to cross-annotate cell types from different study with unique classifiers. So if you have 3 data sets, then you have 3 classifiers, and you try to feed in the data set from basically the data from study 2 and 3 to the classifier that was trained on data set 1. And you do the same thing for the other data sets. And based on this, then you kind of try to infer a hierarchy based on the misclassification. So you could see this starting tree, and then upon the addition of the second data set, some of the cell types are basically mapped to the same level of hierarchy. And then you see this mice2 population at the end of the tree, where there are 2 same, basically there are similar annotations, so mice2, but they were not mapped together. So we further explored this to see why is this happening. And so indeed what we observed was these mice2 cells were species-specific cell states. So these mice2 cells in marmoset were quite different from the one in mouse. And we further validated this. And that's why the model did not put them together. So they're like, if you look at the marker genes, then there are different markers between these two cell states. Okay, I'm gonna get some water. Is there any questions so far? Yeah. No, we build a hierarchy from let's say, depending on where you start. So if you have 3 data sets, then you try to build the data set, the hierarchy based on the first 3, and then you try to update it with new data sets. If you have 1 data set at the beginning, then you build a hierarchy based on 1 data set. Yeah, and the more data set you have, then the more accurate hierarchy you get. Exactly, yeah. And you could also put some prior knowledge here based on the cell ontology there, too. Yeah. Cool. Okay, yeah, so I mentioned cell atlases a lot, but then where the field is trying to get with this effort of building different and huge like references and atlases is, in the end, to look at them like a queryable data set. A queryable entity where you can ask this large cell atlas more abstract questions like, what are the gene programs that have changed between the query and the reference? Which cells have been disrupted? Which cell communication have been disrupted? Or what is the effect of a certain drug? And many other different questions like, what are the genes that are risky for disease? The problem is the state of the art methods for data integration and batch correction or multimodal data integration, they're basically based on, mostly based on neural networks. So you have highly nonlinear methods that you can hardly interpret. And that basically prohibits, or it doesn't allow you to answer many of these questions, because what biologists are interested in, what they actually, so the currency of biology are genes, at least in this case. So the genes and like programs, so they like to explain things in the language of genes. And if you can't give them that, then it's really hard based on some sort of like latent space or unknown variation to analyze the data set. So the idea is, can we go from this non-interpretable embeddings to something which is actually interpretable? Meaning that each latent dimension of these nonlinear methods capture specific variation with respect, the known specific variation. For example, you can have an axis of variation that encodes T-cell differentiation. So if you traverse that latent dimension, then you go along from like not differentiated cells toward like more mature T cells. And the same thing for inflammation. So you have a degree of inflammation from low to high inflammation. And that was our motivation to design a method to basically answer, like a proof of concept to answer similar questions. And what we did was basically trying to encode what we call gene programs. So gene programs are, or many people call them pathways. So a set of genes that encode a biological process or induce certain biological processes. And this is known. So we assume that these biological, these gene programs or pathways are known a priori. In this case, they could be queried in databases, you could mine literature, you could use ChatGPT, whatever, and then, or experts to build a matrix in panel B where we call it gene program matrix. So each row is a gene program and each column is a gene. And 0 means that that gene is part of that, is not part of that gene. So it's like a membership in an indicator variable. And 1 shows that that gene is part of that gene program or not. So the idea was to encode that gene program matrix into a latent space of a conditional generative models, in this case, conditional VAE. So what we did was so there are 3 challenges. A, These gene programs might be noisy, so because they have been built by different groups, so there might be a lot of genes that are not there or that are there, so there could be like both false positive and other case of like incorrect genes in there. B, not all the gene programs might be relevant for your data set, so that's the second challenge. So you have to come up with a way that filter relevant gene programs. And so the first challenge, then once you filter this gene program, also impute the, or like basically add and remove genes within each gene program. And the third challenge is, it's all based on prior knowledge. So you might have some gene programs, some novel variation that is not captured by existing gene programs. So you should go beyond prior knowledge too. And we try to incorporate all these three different constraints into the architecture here. To address the first challenge, so basically allow to impute or add and remove genes within a gene program, we made the decoder linear to actually allow, so we basically build a mask decoder where from each gene program, you only decode the genes that are part of that gene program. So this way we can actually learn a disentangled representation where each latent dimension only capture the variation with respect to specific genes. And in order to allow adding or removing gene in each gene program, so we basically designed a soft mask. A soft mask where you could turn on or turn off a gene based on the data set. The second challenge, selecting relevant gene program. So you could see GP1, GP2, and GPN, so there were those gray nodes, and then we have a GP selection layer. So that's basically like a group lasso sparsity constraint, where the model learns to select or remove certain gene programs. So that's like a selection layer, not just in the level of genes, but rather the level of gene programs. And I'll tell you how we address the third problem, so basically going beyond the prior knowledge. So for the third challenge, so how to go beyond prior knowledge, then we assume that we have a pre-trained model, and now you try to map a data set that might contain novel variations. So there might be some gene programs that are not encoded in the pre-trained model, and you might have some like disease program that is only available and present in your query data set. What we do is similar to the approach that we did with SCRs, but this time instead of adding latent spaces, so you do the architecture surgery as we did it in SCRs, so adding these new nodes to add the new data set. But we also, what we did was also adding a few nodes in the latent space. So having a frozen model, and we basically have some proofs and interesting discussion, empirical evidence here, that if you have a very limited capacity when you train a neural network, then you have like information bottleneck. So the model tries to find novel variations, novel variances that could reduce its loss function. And that's what's happening here. So once we add these gene programs, novel nodes in the latent space, and we only fine-tune those nodes and keep the rest frozen, then the model will hopefully go for what it has to go, yeah? But it's not guaranteed. So in order to guarantee that you find novel variation which is independent from the previously learned knowledge, then you have to enforce independence here. And the way we enforce independence is based on very beautiful theory and a kernel that you can design based on that. So it's called Hilbert-Schmidt independence criteria. So in the end, it's like a kernel that compares the distribution of the newly learned gene program and the previously learned gene program, and then try to enforce independence between this newly learned gene program and the previous one. Yeah. And the previous one. Yeah, so the soft mask is like a binary. We have first hard mask, so we basically put a binary hard mask on the weights of the decoder, and that maps the latent dimension to the gene. So you have a linear layer that all the weights that are not Exactly, yeah. And the soft mask is like the soft version of that, so you're actually allowed to add some genes in there. So I'll show you one example. This is actually published very recently, like 2 months ago in Nature Cell Biology. And here we integrated 5 data sets. So these are PBMC data sets, blood data sets. And then what we did was basically mapping a new data set that contained both control blood cells, so the blood cells that were not perturbed, so normal looking blood cells, and then some cells that were perturbed with interferon beta. So interferon beta is a is a cytokine where it basically induces a big gene expression change. So that happens during immune response, and that changes a lot of genes. And the idea was to map this interferon beta stimulated cells to this healthy reference, and then identify which cell types were more affected and more perturbed and altered by interferon beta versus others. So far you could do all of this with previous methods, yeah? But then you need to do clustering, you need to have an expert, and etc. So the idea was, since we have this probabilistic model, and then we have explainable gene programs, so each latent dimension now is a gene program, so you can perform a statistical test in the latent space and compare the red group versus the blue group. So, and the blue group, you have the blue group from the reference and the blue group from the query, without really worrying about the batch effect, because you actually corrected for the batch effect. So you can perform a statistical test, and there's like a closed form for this. And you can, it's like a hypothesis test. You can ask which gene programs are basically differentially expressed between the blue and the red group. And what you get here is the result of that test in the latent space. So y-axis is basically the base factor that you get from this Bayesian testing. And the x-axis is the rank of different gene programs. So the top 2 gene programs are, as expected, related to interferon signaling. So that's where interferon beta is basically affecting and altering. Then you can actually visualize these 2 gene programs in the latent space because you know them. And if you just collapse the data into these 2 dimensions in the latent space, then you can see that the variation is super clear. So you have the group of red cells, and then you have the group of the blue cells. No matter whether they're from the query or the reference, then you see that these 2 groups are separated. And this is not cell type specific test, so this is just across all the cell types. So these variations are differentiating all the cell types in interferon group versus all the cell type in the control group. But you could do this also in a cell type specific manner. So you can perform these tests for individual cell types. So to basically extract cell type specific gene programs. So you could see like 2 gene programs that separate B cells from the rest. And the same for other cell types. So if you combine now these 2 analyses, so cell type specific gene programs versus perturbation specific gene programs, so all the gene programs that are relevant for interferon signaling, then you get and then you have activity for each single cell, yeah? So you basically can tell this cell is active with this degree in this gene program and so on. And what you see is a Violin plot of different cell type across 5 different gene programs, across 2 groups, interferon and controls in the x-axis. So the first 2 gene programs, interferon signaling, interferon alpha beta signaling, that's the first one that I showed. They're actually activated across all cell types. So that's like a global response of interferon signaling that happens across all the cell types. So you could see in all of the cell type, interferon beta is basically upregulated versus the control cells. And then you have some cell type specific gene programs. So interferon has this selective system. So it basically affects some cell types in a different manner. And in this case, what we identified was a regulation of metabolism in the myeloid cells that we could basically identify using this method, and this was not reported before. Okay, so I'm going to skip this one, but this is just one example that you can apply this this method on also patient data, and identify why certain patients don't respond to certain drug, and identify the gene programs that might be responsive for why the drug is not affecting in those patients. That could potentially give you some targets for the next drug that you want to repurpose or redesign here. Okay, I'm going to skip this. And one interesting part, we're more like in a technical part, is, yeah. Mm-hmm. Yeah, exactly. I mean, of course, you need an expert to then annotate it if you learn a new gene program to see what it's actually doing. Yeah, based on the genes. That's just a hyperparameter. It's a hyperparameter, and yeah, it's not an easy one to tune because it depends on how different the query dataset is from the reference. So if you have really different dataset in the query compared to reference, then you might allocate a lot of nodes there. But if you have a very, it's like a slight difference between the query and the reference, then 1 or 2 nodes might. I mean, we have a default which is, I think, 5, but it could vary across data sets. Yeah, this is completely unsupervised. So the model is not aware of. There are some identifiability issues here, but since we have a linear decoder, then it's easier to tackle those. Yeah, so we have some proof and theorems in the paper itself. Check it out, please. One last thing here is that so in this model that I showed, you had a nonlinear encoder and a linear decoder. So you have a low capacity versus nonlinear encoder and nonlinear decoder. And what we showed in the paper is adding prior knowledge to the model actually can improve the performance of this model versus nonlinear encoder and nonlinear decoder. So the class of models with higher capacity in learning. And that is because you have a lower variational gap between what you learn, what the true posterior and the one that you learn in the class of models where you have like nonlinear and linear decoders. So that's what you see on the right. If you have, so it's called XPMAP, what we have, and we have SCVI, which is like gold standard, where you have nonlinear encoder and nonlinear decoder, but it does amortize inference. And then you have SCVI non-amortize, which is basically a more accurate way to infer latent variable. And you could see XPMAP, which is a different class of model and obviously a weaker class of model compared to those two, performs on par with the existing one. And if you do non-amortized, then you can actually get a better performance. So it actually helps with the optimization and also the generalization, which I show on the left, but I'm not going to go into detail. So adding prior knowledge here can actually improve the generalization of ML models, at least in this case. Okay, so I have half an hour. And yeah, so the next question is, so whatever I showed so far was basically identifying cells, cell types, and gene programs. But I'm going to talk about 2 methods that actually go beyond cell types and then look at the population level variation. What we're interested in this setting is to have an embedding for cells and also having an embedding across patients or samples. So you could basically look at both of these embedding and see how at the patient level things are varying and how at the cell level things are varying. So this question will allow you to answer 2 things. So population level. And I introduced 2 different models for this. Where is my population? Meaning that what is similar and difference across samples? And then where is my phenotype and disease? Which cells are responsible for 2 different samples to be different or similar to each other? So the first model this is actually a preprint, but it will come out in Nature Methods soon. It's called single cell population level integration. It's called SCPOLY. And then this is based on conditional VAEs. But in this case, instead of encoding the batch variables using a one-hot encoding, so what we usually do is for each batch variable, you have a indicator variable, like a one-hot encoding schemes. And in this case, we encoded samples and samples and the batch in a continuous space where we basically have a learnable embedding for each patient or each sample. Then you have like a continuous space of patients and samples. And to further model cell types, then we leverage ideas from meta-learning, prototypical learning, where we try to learn a centroid. So this model is aware of the cell type. So it's like a semi-supervised model, where the model is now aware of the cell type level variable information. So basically you learn a centroid and you try to push all the cells that are from the same cell type to the centroid, similar to how you learn k-means and etc. So, and then at the query time, so if you have a new data if you want to apply SC-arches in this case, then the query mapping technically, this is a very technical and engineering thing, but like, I really like it. So, you don't need to optimize those, because once you want to implement this architecture surgery, you have to mask the right weights, and then you basically have to use hooks in PyTorch, and hooks are expensive. I promised myself not to talk about it, but I like it. So, and in this case, since you don't need to use hooks, and you have this conditional embedding. So you have a dictionary. Whenever you have a new data set, you basically add one element to that dictionary, and you only optimize that embedding corresponding to that patient or sample. Okay. And then related methods are another method from like Nir Yosef called Master VI for population level variance. But I'll show you one example. So first, you can do all the single cell type level operations. So you can build a reference, and so reference building. So this is a reference building method, but it could be combined with reference mapping methods too, like SC-arches, yeah? And what you see in the third column is the cell type awareness of this method. So you basically learn a centroid, and those dotted, so those circles are what the model actually learned on the latent space for each cell type. And then you also have some unannotated, so those smaller circles, which are basically unannotated prototypes to further cover the latent space and learn a better representation. And once you have the query data set, then you actually learn new prototypes for the query data set. And what you see in the last column, second row, is basically the study level embedding. So those are the continuous embedding that we're optimizing for each sample during the training. In this case, you could see that the model identified that like two droplet-based technologies are similar. So they basically, they're embedded closely to each other. And then you have, so this is merely for like technical variability. But I'll show you, yeah, so these are some benchmarks that, it works better than previous data sets on both integration, previous model based on integration and also cell type annotation. Another interesting part is all these prototypes that I showed, those circles, they could be used for cell type transfer. So if you have a query data set, then you can map it to the latent space. And then based on the anchors, based on the distance, then transfer cell type annotations from the reference to the query. But they also have a good behavior and like very interesting behavior that you can use for inferring uncertainty. So if you map a new data set to this reference, then the distance to the prototype that was used to annotate these cells could be considered as some sort of uncertainty. So basically distance-based uncertainty. And we indeed show that, this is a similar example that I showed in the beginning of my talk, that you have a k-NAND classifier to infer that was uncertain, but this time we actually learned this uncertainty-aware model doing the inference, and we showed that it's better than existing simpler way of doing uncertainties. Okay. So now I'm going to show you one example that we integrated 25 PBMC datasets to learn a population level, sample level embedding across all these 25 different datasets, which are basically from 2,321 different samples, so individuals. And so the first iteration was 8 million cells almost, and now we basically extended to like 12 million cells. And so on the top, you'll see the cell latent space. So each dot is a cell. And at the bottom, so in the second row, each dot is a sample. So this is like a sample level embedding. And we colored sample level embedding based on different metadata. Dataset, assay, disease, and you could see the variation is across, so we had like multiple states here. So there were like disease cells, there were healthy cells, multiple diseases. And when you go to this regime of like many data sets, then disentangling between the technical and the biology is challenging. So you could see that. For example, all the cells that are healthy, they're actually profiled with 10x3 prime version 2. So that's like a different chemistry. And at the same time, some of them are actually combined with cells that basically came from lupus patient. On the other side, all the cells they're basically they were like COVID-19, influenza, that had similar immune response almost, they were embedded close to each other. So now the question was, how can we disentangle this variation? Is it batch effect? Is there like a disease effect here? What we did was correlating the latent space of the sample embedding with the gene level embedding. So basically identify how the sample level embedding changes with respect to the gene. So we took the PCA of the latent space of the sample level, and we correlated that PCA with gene level representation. So the PCA1, so the PCA is basically, the PCA1 is like the x-axis, and that's where the disease and the healthy ones are separated. And the genes that were mostly effective here were the genes that were related to immune response. So the difference between the healthy COVID and influenza. At the same time, there were like PC2, which was the separation between different technologies. It was affected by many genes that were encoding ribosomal genes. So those are the technical variation what usually when you analyze single cell data sets that come from like batch effect. So in this case, both biology and so biologically meaningful signals that were related to immune response and the difference between healthy and disease were convoluted with batch effect. But you could actually have a way to disentangle these two things. But I think, and this is in revision, this is, we have a revised version of this. And there is actually like a more automated way to analyze this. But yeah, this is how it looks like with like many thousand patients. Okay, so I'm gonna skip this one. But the idea is you can use this patient level embedding to actually map new patient to this level. So what I showed so far was mapping cells to the cell level space. You can actually do that for the patient too. So to this patient space, then you map new patients, and then you can train a classifier on the patient space to annotate new patient in terms of disease severity, whether these patients are diagnosed with a severe COVID or moderate COVID and so on. Okay. Is there any questions so far? Yeah. Oh, the model is not, yeah, so what do you mean exactly? So, oh, yeah. So we use a very high level cell type annotation. So basically, yeah, lymphoid versus others, so not super detailed to not bias the latent space towards specific lineage. Yeah. Cool. So whatever I showed here was mostly based on single-cell RNA-seq, but I just want to show that like all of these methods are all of these methods are basically extendable to multiple modalities. And this is a work from, again, my PhD, but in collaboration with Anastasia, who's also I'm co-supervising at the moment, where we designed a method to encode partially overlapping multimodal data, meaning that you might have a single-cell RNA-seq data, a single-cell chromatin accessibility data, and a data set where you have both chromatin accessibility and single-cell RNA-seq. Can you integrate this or not? So in this case, we learned a generative model. So you could see different modalities. So we have like different encoders, and then we learn a generative model based on the concept of product of experts. So I'm not going to go into detail here, but like it basically allows you to learn a distribution where you can map unpaired data, meaning that if you have some missing modalities, then you can statistically model this and map it into the latent space. That also allows you to integrate multiple modalities. But I refer you to the paper. What it allows you in the end is to build a multimodal reference. So you can integrate across all of these multiple modalities, and then combine with reference mapping methods, then you can actually do multimodal query mapping, meaning that if you have a data set where you have some missing modalities, you can still map it into the reference. And finally, you can also infer a missing measurement based on a multimodal reference mapping. So similar to the site-seq example that I showed you, that you can impute the missing proteins, but you could actually impute missing chromatin accessibility, for example, if you have a single-cell RNA-seq listed alone. This is one example that we built a trimodal reference atlas. So in this case, we had a site-seq data set and a multi-ohm data set. So in a site-seq, you have protein abundance, single-cell RNA-seq, and with the multi-ohm, you have single-cell RNA-seq and chromatin accessibility. So we basically integrate all of these three different modalities to build a reference on the left, and then you can query this reference with different type of modalities. So you could see we queried with the single-cell RNA-seq alone, a site-seq alone, or just the chromatin accessibility, so ATAC alone, and all of them map quite nicely to the dataset. But going beyond like chromatin accessibility and site-seq, there are other single-cell technologies that are multimodal. For example, you can capture the gene expression of T cells plus their T cell receptor. And so T cell receptor, or TCR, is basically the way that T cells identify antigens. And in this case, there are measurements where you can combine single-cell RNA-seq and TCR measurements, so you can capture what is the gene expression for this T cell and what type of antigen, and what is also the T cell receptor for this T cell. So the T cell receptor, the two most important part of it is basically alpha chain and beta chain. And these are the radars basically where it identifies antigens. And these are sequences, yeah? So in this case, we have gene expression, and then we have two sequences. And in this case, we combine, so we modeled sequences using transformers. And in a similar fashion, we combine it with gene expression, and we have a mixture module that combine the T cell receptor information and the gene expression information. This is a collaboration with Benjamin Schubert in Munich, where we basically show that having two modality is actually better. This might sound obvious, but it's not obvious that like if you actually combine multiple modalities, then you can get a better representation than the single modalities. So it's not always the case. And so in this case, we compared three different representations. A model that were only trained on TCR, a model that was only trained on RNA, and a model which we call MVTCR, multi-view TCR, where it combines both of them together. And they are colored by antigen specificity. So those are basically the peptides that the T cell would basically attach to. And you see that in the right representation, so the one with MVTCR, the ones, so for example, those two clusters, three clusters that I highlighted, they were grouped together compared to the RNA or the TCR one. And we actually show it. If you cluster the RNA cluster And then you map those clustering to the specificity labels, so as you see on the top. And then you see that like more coherent clustering in the joint model compared to the single model. And the same thing for the clonotypes, yeah? And so this is in review, and one of the reviewers actually asked about, okay, this is good, but then so what? Like which modalities are actually contributing here? So we came up with a way to decompose the modality contribution. In this case, you could see that cluster that was basically put together by MVTCR compared to the unimodal models. It has the highest score on the TCR. So basically the TCR modality, the sequence modality contributed more here compared to the gene expression. And that's why the model actually learned to put this cluster together compared to unimodal models. And if you look at like different cell types, then you can decompose the contribution of different modalities here. Okay, so this is again another vignette here where we apply this model on a data set where we have data from different patients of COVID. And the idea is, in this case, we want to separate two classes of T cells. So COVID-19 specific T cells. So those are the T cells that actually identify the COVID virus and antigens from that. And the class of T cells called bystander cell. This is actually like a big question in immunology where you want to identify separate between bystander cells and the specifically activated cells. So bystander cells, they are activated, but they're not activated through antigen binding. So there was no antigen bound to this T cell receptor. So they actually were activated and showed a similar signal to the activated cells based on this. And what we show that if you combine these two modalities, then you can actually find cluster that separate bystander cell versus COVID-19 specific T cells. Okay, so I have like 15 minutes, right? So I might skip this one, but I wanted to show you one more example on the population level data sets where you can actually integrate multiple patients. This is actually going back to the second question. If you have a population level data set, can you learn a population level embedding and tell what are the cell types and cell states that actually separate one patient from another one? Yeah. And so this is the architecture of this model. So we call it like multi-mill. So it's based on a concept of multi-instance learning. So we integrate across multiple modalities, but then we have a classifier that actually, because when you have a data from a patient, you don't have labels for individual cells because you don't know whether they were affected by disease or not, but you have the label for the whole patient. Yeah. And in this case, what the model does is basically identify which cells and cell types are actually responsible for that patient to be classified as, for example, like a patient with malignant cells or primary one. Yeah. So in the end, you can get a single cell resolution map for each patient where it basically picks up cells that were responsible to annotate that patient to a specific disease severity. Okay, I'm not going to go into detail here. I'm going to skip to the last part. No, this is the second to last, but this is the third part where this is my favorite line of research, basically predicting perturbation at the single cell level. The idea is, can you design a machine learning algorithm that receives a single cell and a drug or a genetic manipulation and predict how that single cell actually responds to that perturbation? And why is it useful? Because this is actually an active area of research in drug discovery. So people are moving from target-based drug discovery to phenotypic-based drug discovery. So it means that there are complicated diseases and you don't know what are the targets, what are the genes responsible for those. So you can't target them with a drug. So what do? then you treat them as a phenotype, meaning that you have a representation for the healthy, representation for the disease, and you try to basically come up with a drug that pushes the disease cells toward the healthy cells. And this is only possible if you have high resolution information about the disease, meaning that you need to have single cell type level information about the disease. But single cell technologies are expensive, so you can't do like 10,000 drugs and like, brute force the hell out of the whole thing. And so what you need to do is basically design a very rational experiment where you only select a specific drug and you apply it on your data set. So you need in silico drug design or in silico experimental design. But the first question is, can you actually, model this? Can you predict how the cells would respond to unseen drugs? This was like, again, part of my PhD. So I had like, I think we were the first group basically addressing this question at single cell level. And there are a series of publications here. But in the end, the ultimate goal is basically to design to basically explore the combinatorial space of perturbation and design a better experiment. And in order to do that, then you need to have a model that can perform a counterfactual prediction, meaning that you give it the cell and you ask the model what would the cell would look like if it was treated with drug A or drug B. Yeah. So the first method that we designed for this was based on a super simple method based on vector arithmetics, which was basically inspired from imaging field. At that point, people were crazy putting glasses on things, and now it doesn't happen anymore. But like, so basically manipulation on images and etc. So the same concept could be applied here, that you have a disease cell and you have a cell which is healthy. So you learn the difference between these two in the representation, and then you add this difference to the cell which were not basically measured with the disease, and you can predict the gene expression for that. And later in the second iteration, we formulated this as a distribution matching problem. If you take the distribution of the healthy cells and the disease cells, can you learn a mapping to match these two distribution? If you learn this mapping, then you can actually push cells from one distribution to another. The second one. And this was basically my work during, so my internship at Facebook AI, where we actually try to make this whole thing more interpretable, like a linear model. Can we decompose these effects such that we have the overall expression decomposed into a basal state, meaning that this basal state is free of the variation of perturbations and cell type, and you decompose the gene expression into basal state plus variation to the cell type, variation to the drug, and so on and so forth. Yeah, and this is basically what we call CPA, compositional perturbation autoencoder. This is a generative model where the generative process of the data is actually a composition, meaning that you learn a basal state, you learn a perturbation state, and then you try to reconstruct the whole thing by a compositional formulation. So this is the architecture, but like I'll skip that. What it does basically during the training, it learns how to disentangle effect of perturbations, so cell A, from those two drugs. So you learn a basal state, cell type level representation, and the perturbations, and it tries to basically put them back. Yeah? And then reconstruct it. And during the testing, then you have counterfactual prediction, where you have cell A treated with two drugs or two perturbations. You try to separate those, and then you have cell B that were treated with three drugs. And then you can ask the model what would cell A would look like if it was treated with three drugs. And this is a very like basic simulation on the same application of the model on the same data set from like interference signaling versus control that I showed before. So in this case, you have different cell types and two groups of perturbation. Control, so non-perturb, and stimulated with interferon beta. That's like a chemical that I introduced, yeah? So this is the latent space of the model if you only provide the perturbation. Meaning that you ask the model, learn a composition that can learn a basal state that is free of the perturbation. And this is the representation of the basal state. So it learned to correct for the perturbation. And this is the latent space. So in the reconstruction, it adds back the perturbation. And if you give it the second covariate, so if you ask the model, correct for the perturbation and correct for the cell type effect, then you get this almost like noisy-like state where it actually corrected for both. So the first cell line that we applied it was, the example was basically a highly multiplexed cancer cell line, 3 cancer cell line. So we had like 3 cancer cell line perturbed with 188 drugs in 4 different dosages. So in overall, you had like 500,000 cells and 4,500 unique conditions if you combine dosages, drugs, and replicates, and etc. Since you actually learn a latent space for perturbation, so this is separate from cells, you can actually learn a perturbation space where it captures the similarity across different drugs. So if 2 drugs induce similar phenotypes, these 2 drugs will be embedded closely to each other in this perturbation space. So you could see this perturbation space, and you could see like drugs with similar mechanism of action, mode of action, basically like epigenetic regulation. These are the drugs that are tyrosine kinase inhibitors. They're closely, tyrosine kinase inhibitors and also epigenetic regulation that change the epigenetic are closely embedded to each other. And then you have lots of drugs that are basically not effective. You have the same thing for the cells, so the cell lines. In this case, they were 3 different cancer cell lines, so they were quite far from each other. And so we trained this model on this data set, and we used this perturbation space to design a second experiment. In this case, the second experiment was supposed to be a combinatorial experiment, meaning that we wanted to identify the drugs that were combined, so like a cocktail of the drug that were effective. So we used this perturbation space to design a second experiment where we basically had 60,000 cells across 32 mono and combination of drugs. So this is the newly generated data set. And using this method, then we actually showed that the model can actually predict unseen drug combinations. Better than, at that point, there was no method doing this, but there are like a wave of methods that are doing this now. And then we extended this because at that point, we couldn't actually predict completely unseen drug. We could have predicted, if you have drug A and you have drug B, you could have predicted A plus B, even though the model hasn't seen that. You could have predicted A, and if you haven't seen B at all, then you couldn't predict A plus B. So we had to incorporate a representation for the drug itself in order to completely, to basically predict completely unknown drugs, so out of library drug. This is a recent publication, not recent anymore, so this is like last year publication in NeurIPS, where we extended the architecture of CPA to incorporate chemical priors here, which is called ChemCPA. And then we actually designed a transfer learning approach where we combined both single-cell RNA-seq and bulk RNA-seq to basically compensate for the lack of data in single-cell RNA-seq screenings, and that actually shows better performance. And the same thing, if you apply this model on not drug screening, but rather genetic screening, so perturbations that are based on CRISPR knockout or CRISPR or overexpression of a certain gene, you could still learn, in this case, gene functions. So the perturbation space that you see encode, so it basically clustered genes that were doing or inducing similar phenotypes in the cells. And then we showed that you can predict unseen gene combination from different epistatic or like synergistic interaction. And then we imputed, so this was an example where we imputed 5,200, I guess, 329 missing genetic combination from the initial experiment that they couldn't capture all of these combinations because it was too expensive for them. So we in silico generated 50 cells per combination, and then we show that you can actually use the uncertainty to identify relevant next perturbations. And this was published recently, so you can check it out. It also went on the cover. And let me know if you have any question. Can I have 5 more minutes, or? Okay. This is like really out of open. Yeah, so all of these perturbation modeling, you could actually extend them not just to work on genomics level information, but rather on images. So in this question, we were interested to see whether we can design a generative model that receives the image of a cell. So you have the morphology of that cell, and then you have a drug. And the question was, can the model generate how the cell would look like after treatment with the drug? So I'll skip so we designed a generative model to perform this task, and what it does is basically quite interesting. So it works on cell painting assays. So this is another assay that you could do phenotypic drug discovery based on that, meaning that you don't get the gene expression anymore, but you get the morphology, and people use it actually to identify active drugs. But again, the same problem here. You can't really, screen all possible drugs in the world, yeah? And this is a generative model that allows for that. And the performance compared to existing approaches, state-of-the-art approach, so we basically observed like an 85% improvement on the task of unseen drug perturbation. Okay, so I had a few stuff on the spatial transcriptomic, but then I skipped that. Yeah. With this, I conclude my talk. So we have other work on dynamic modeling or modeling populations and other things, but I didn't show those. So if you're interested, oh yeah, in the end, there were like work of a lot of people that I collaborated in the last 2 or 3 years. And yeah, and then there are like available positions. If you're interested in this line of work, please reach out. Yeah, happy to take questions. Thank you.
Okay, questions?
Yeah.
I'm going here.
Hey, thank you. Really amazing talk. I just want to stop a bit in this fundamental question you made regarding when is good using multimodal data. More data is going to be better, when it's not. So for all the research that you have described, can you tell us a few insights on that? When did it work? And well, you showed up when did it work, but when it didn't work.
Yeah, that was one of the rare cases that it actually worked. So there are like tons of multimodal data integration, but in the end, if you dig deep into them, you can't really, yeah, unfortunately, you can't really, see novel phenotypes by combining, at least at a single cell level, as far as I'm aware. There are use cases where chromatin accessibility and gene expression combined together are helpful, because then you can dissect some progenitor cells based on chromatin accessibility that are not possible to basically to be resolved with single-cell RNA-seq. So there are very few use cases in that line of work, and now we have it for the T cells. But for the other things, I'm a bit doubtful about what is going on.
Hi, thanks for the great talk. I'm quite interested in the last part, which is the cell imaging part.
Yeah.
So it looks like from the schematic figure is that you are trying to compare between the cells after the drug, the treatment of the drug, and compare it to the original look. So I'm curious that do you also considering that to compare in a larger scale, like to see how the distribution of the cells will change after the drug treatment?
We do actually have that. I skipped that part. So here, yeah. So these are the cells that we generated using this model. Then there is this software called Cell Profiler. I don't know whether you heard it or not. So that's basically what people actually use to analyze a cell painting data set or imaging assays. So that basically is like more like a manual feature extractor. So if you give it image, it will give you sort of like, based on like cell size and etc., some interpretable features. And what you see here is we generated cells for drugs that we haven't seen, and then we looked at the features and based on cell profiler, and we basically compared the distribution of these features versus the ground truth features. And then you could see here basically second row that I think it's not super clear, but like the dark red versus like the light red, they are aligning on the top of each other. And that's basically the in silico generated versus the real ones on the feature and the cell profile level. Hope that answered your question.
Any more questions?
Hi, so this is a question about the VAU where you had the linear decoder.
Yeah.
So you had a kind of a fairly constrained linear decoder.
Right.
What was the coverage like? So I don't know what the source was, like gene ontologies or something like that.
Oh, yeah. So we had different databases. We used Reactome, we used PangLao, we used some manually curated gene programs that we built based on, in collaboration with experts. So it was a combination of those. So in the end, we had like 1,000 gene programs or something. And then after filtering, it was like 200 or something.
And did those cover all of the genes that you were trying to like reconstruct?
Oh, okay. So coverage on the gene level. There will be some genes missing. Yeah. So that is one challenge here. Yeah. And there is like another challenge, which is basically these things are hierarchical, so you need to know where to cut in these ontology-based stuff. And so there are some problems there. Yeah.
So did you observe that even maybe the reconstruction loss is a lot worse? The clustering still seemed to be, or however you
Right. Yeah, so we didn't indeed evaluate on the reconstruction loss. So we took the latent space as a proxy for reconstruction, because if you fail the reconstruction, then you can actually fail the latent space if you can separate the two things. But like that's a valid point. So we didn't do it in the end. Yeah. But it should be weaker than that.
Maybe time to get recaffeinated. So let's thank Mo once more.
Thank you.